Imagine for a second that you are tasked with mapping every single star in the observable universe. But there's a catch. You have to do it using, I don't know, thousands of different telescopes scattered all over the Earth.
Right.
And absolutely none of these telescopes are calibrated the same way. The data formats are completely different. The lenses are different. And the scientists operating them aren't even communicating with one another. It sounds like a total logistical nightmare.
It really is.
Well, that fragmented, kind of chaotic reality is exactly what microbiologists have been dealing with when it comes to mapping Earth's microbes. Until now.
Yeah, it really has been a massively disjointed effort for decades. We have been collecting this incredibly rich sequence data from every conceivable environment. I mean, from deep ocean thermal vents to the highest alpine soils, but piecing those distinct data sets together into one coherent planetary picture has been practically impossible. The data was just too noisy and too siloed.
Which brings us to the core mission of our deep dive today. We are unpacking a truly monumental resource published in the journal Cell in 2026. It is called the MicrobeAtlas. And this project actually manages to unify over 2.4 million microbiome samples from all across the globe into a single, searchable, standardized map of invisible life.
Yeah, exactly. And to give you some context on the source material we are working with today, we are diving directly into the primary scientific paper detailing the creation, the underlying bioinformatics, and the initial global discoveries of this database.
Right.
The researchers took an unprecedented volume of publicly available sequencing data and harmonized it. They built a tool that allows us to track microbial lineages across diverse environments, climates, and hosts with a level of resolution we just haven't had before.
It's essentially Google Earth, but for bacteria and archaea. So here's how we're going to navigate this with you today. First, we're going to break down the mechanics of how scientists actually identify and track these invisible ecosystems, especially when the historical data is so incredibly messy.
Yeah, that's step one.
Right? And from there, we'll map it out and look at the surprising geographic and evolutionary trends that emerged. And finally, we are going to expose the massive blind spots in our current scientific knowledge, some of which fundamentally challenge how we conduct medical research.
It's a progression that really forces a paradigm shift in how we view the biological machinery of our own planet.
Absolutely.
Yeah.
So let's start with that problem of fragmented data. For years, the status quo was simply that different labs use completely different processing pipelines. Before MicrobeAtlas, if you wanted a unified global database, the gold standard was the Earth Microbiome Project.
Right, which was a great project.
It was highly respected,
Yeah.
but it only encompassed about 27,000 samples. MicrobeAtlas scales that up to 2.4 million. So how do they achieve that kind of scale? I mean, if we were moving away from traditional lab cultures, how are we identifying these microbes from environmental samples at this volume?
Well, we have to look at the evolution of the sequencing technology itself. We obviously know that trying to grow environmental microbes in a lab culture is a losing battle.
Because they just die.
Yeah, most of them simply refuse to grow outside their highly specific, complex, natural habitats. You can't just put them in a petri dish. So the field largely moved to culture-independent methods. A very robust approach is building MAGs, or metagenome-assembled genomes. This is where you extract all the environmental DNA from a sample and computationally try to assemble the entire genomes of every microbe present.
But doing that at a planetary scale sounds computationally terrifying. I always picture MAGs like, taking 1,000 different jigsaw puzzles, throwing all the pieces into one giant pile on the floor, and then try to put them all back together without knowing what the pictures on the boxes look like.
That is an incredibly accurate way to visualize it. It's a massive puzzle. And because it is so computationally expensive and complex, doing that for 2.4 million samples just isn't feasible yet. So to build MicrobeAtlas, the researchers utilized marker gene sequencing. Instead of trying to rebuild the entire genome, which, following your analogy, is like doing a full exhaustive background check on every single microbe scientist, just look for one highly specific molecular ID badge.
Yeah.
The ID badge, right?
Exactly. In this case, it's the 16S SSU RNA gene.
I want to dig into that mechanism for a second because we hear about 16S all the time in microbiome literature. Why this specific gene? Why does every single microbe wear this exact ID badge?
It comes down to evolutionary biology. The 16S RNA gene codes for a critical component of the ribosome,
which makes proteins.
Right. It's the cellular machinery that makes proteins. And because protein synthesis is absolutely essential for life, the structure of this gene is highly conserved across all bacteria and archaea. You can't mess with it too much or the organism dies. However, and this is the genius part, while the core structure remains exactly the same, allowing us to easily find it, there are specific hypervariable regions within the gene that mutate at a very steady rate over evolutionary time.
So those variable regions act like a barcode. You find the universal frame of the gene, but then you read the variable middle part to see exactly who you were dealing with. It's cheap to sequence, it's incredibly fast, and it lets you see who is at the party without needing their entire life story. Exactly. And because this 16S marker has been the standard for years, there are literally millions of these sequences already sitting in public archives. It provided the perfect common denominator to link thousands of disparate studies together. But relying on this ID badge requires us to address a crucial piece of terminology used throughout the database, and that's OTUs, or Operational Taxonomic Unit.
Right, because if you're only looking at a barcode, defining a strict species gets complicated. Microbes are constantly swapping DNA through horizontal gene transfer, so the classic definition of a species kind of falls apart, doesn't it?
It does. The lines are incredibly blurry. You can't just call them a species in the way you'd call a bird a species. So instead of rigidly trying to assign a classical Linnaean species name to everything, scientists group microbes by their genetic similarity into these OTUs. If two microbes have 16 SID badges that are a 97% genetic match, they are grouped into the same 97% OTU.
I always find it helpful to think of OTUs like a postal code system.
Oh, that's a good way to look at it.
Yeah, like if you group microbes at a 90% OTU match, that's a pretty broad resolution. It's like knowing a microbe lives somewhere in New York State, you know, the general family. But if you increase the stringency to a 99% OTU match, that is highly specific. It's like knowing that microbe lives in a specific apartment building in Manhattan. And what's fascinating is that the MicrobeAtlas tracks over 650,000 of these hierarchical OTUs.
Yeah, the adjustable resolution is what makes the tool so analytically powerful. A researcher can zoom out to observe broad phylum-level dynamics across an entire ocean, or they can zoom in to track a very specific 99% identical strain of bacteria as it moves from an agricultural soil sample into the gut of a livestock animal.
Okay, so let's do a quick mini recap here for you listening. We started with a mountain of messy, disconnected data. The solution was using the 16S ID badge to identify who was there, and then using the OTU postal code system to group them by genetic similarity. This allowed researchers to unify those 2.4 million samples. So here's a question for you to consider. Have you ever thought about the sheer volume of data sitting in public archives right now, just waiting to be standardized so it can actually solve major problems?
It's a staggering amount of untapped potential, which perfectly transitions us into the logistics of how they actually built this atlas. Moving from raw data to highly defined, standardized ecosystems was an absolute engineering marvel.
The data mining required for this planetary scale map is hard to wrap your head around. The researchers accessed the NCBI Sequence Read Archive, which is this massive public repository, and downloaded 707 terabytes of raw data.
Just a massive server pool.
Yeah. They searched for projects tagged with keywords like metagenomics or microbiome and just scooped up over 214 billion microbial reads. But you can't just slap 707 terabytes of raw, unverified data onto a map. How did they filter out the noise?
The filtering pipeline was immense. They utilized a high-performance bioinformatic tool called MAPSEC. This isn't just a simple text-matching algorithm. MAPSEC aligns sequences against a reference topology and calculates rigorous confidence scores. They had to discard low-quality reads and sequences that were simply too short to provide a reliable ID. Crucially, they had to filter out lab contamination, which is a massive issue in legacy sequence data.
That makes perfect sense. Because if a researcher sneezed in a lab in Paris while preparing a deep-sea thermal vent sample, you absolutely do not want the atlas showing human respiratory staphylococcus thriving at the bottom of the Mariana Trench.
Precisely. MAPSEC's confidence algorithms help flag and isolate those incongruous sequences. After this rigorous computational gauntlet, that initial 707 terabytes was distilled down to 1.15 million pristine, highly comparable community profiles. These are samples where the researchers can confidently state exactly who is present and in what relative abundance.
But knowing who is there is completely meaningless if we don't know where they are in a biological context. A GPS coordinate alone doesn't tell you much. If I give you a latitude and longitude, it could be a forest, or it could be a paved parking lot right next to that forest. How do they give these 1.15 million profiles a meaningful ecological address?
This is where the paper makes a massive leap forward. The creators didn't rely on the subjective labels provided by the original scientists, who might have just labeled their sample as dirt or soil. Instead, they computationally linked the physical GPS coordinates of these 1.15 million samples directly to the IUCN Global Ecosystem Typology.
The IUCN being the International Union for Conservation of Nature.
Correct. By integrating the GPS data with the IUCN framework, the MicrobeAtlas effectively maps that microscopic DNA to 34 distinct macroecological functions. So we are no longer looking at generic dirt. We can specifically compare the microbiome of tropical savannas in sub-Saharan Africa to pyrrhic tussock savannas in Australia. We can evaluate cryogenic zones against temperate boreal forests.
It effectively bridges the gap between the microscopic world and the macro world we can actually see and interact with. It puts microbes on the exact same ecological map that conservationists use to track elephants, deforestation, and coral reef bleaching.
And they applied this uniformly. They mapped over 65,000 terrestrial samples to these highly specific ecological functional groups. For the first time, we can clearly see how global vegetation patterns and massive climate systems and anthropogenic impacts directly shape the invisible microbial layer of the Earth.
Time for another quick mini recap. The team cleaned up 707 terabytes of raw data using that MAPSEC tool to get 1.15 million pristine samples. Then, they linked those samples to 34 macro ecosystem maps using the IUCN framework. So my question for you is, if you could drop a pin anywhere on a map to see the invisible life thriving there, where would you choose? An Arctic glacier? A rainforest canopy?
The possibilities are literally endless.
Right. So we've taken billions of messy reads, filtered them down to a pristine data set, and anchored them to macro ecosystems. When you finally drop all those meticulously clean pins onto a global map, what do you see? This brings us to the aha moments of the research, and one of the most interesting findings relates to latitudinal diversity gradients.
Yes, the equator effect. In classic macroecology, it is a foundational rule that you find significantly more animal and plant diversity in tropical regions near the equator than you do at the extreme freezing poles. The environment is more stable, there's more solar energy, so biological complexity explodes. But for decades, there was a fierce debate about whether invisible microbes followed this same rule.
Because the old assumption was always, what was it? Everything's everywhere, but the environment selects. The idea that microbes are so small and blow around the globe so easily that latitude shouldn't matter.
Exactly. But the MicrobeAtlas data decisively settles this. The data reveals a resounding yes to the equator effect for microbes. Marine microbial diversity absolutely peaks right near the equator, specifically between 0 and 20 degrees north latitude, before tapering off as you move toward the poles.
And it wasn't just isolated to the oceans, right? The terrestrial data reflected this too.
Yes. When they analyzed sufficiently sampled terrestrial biomes like diverse soils and global deserts, they observed a highly similar diversity spike in the lower latitudes. It proves that microbes, despite their microscopic size and adaptability, are bound by the exact same planetary and climatic constraints as a rainforest tree or a tropical bird.
That fundamentally connects the micro and macro in a really satisfying way. But the map also highlighted areas of profound ignorance, what we might call the dark matter of the microbiome. The atlas clearly showed that soil and aquatic microbiomes are absolute hotspots for uncharted, entirely uncharacterized microbes.
If we contrast the environmental data with animal and human microbiomes, the picture is completely flipped. Biologically speaking, the human gut is actually far less diverse overall than a single scoop of untouched forest soil. But because biomedical science has poured billions of dollars into studying ourselves, the microbes in the human gut are incredibly well documented. The true dark matter of biology, the vast reservoirs of undiscovered genetic potential, is sitting in the dirt beneath our feet and in the open oceans.
Which brings us to an insight from the paper that I found endlessly fascinating, and that is the distinct survival strategies of microbes. The researchers categorize them into generalists versus specialists, or as I like to visualize them, the rugged backpackers versus the delicate homebodies.
It's a very apt visualization. The data distinctly clusters these two evolutionary pathways.
Think of specialists as the homebodies. They only thrive in perfectly climate-controlled, highly specific environments. For example, the paper points to the Lactobacillus family. It's a specialist that absolutely loves the warm, nutrient-rich, anaerobic environment of the mammalian digestive tract. Or Funcibacter, which is a specialist almost exclusively found hovering in specific strata of freshwater lakes. They know exactly what they like, their biological tools are hyper-focused for that one job, and they do not leave. Generalists, on the other hand, are the backpackers. They can survive almost anywhere. The paper highlights Pseudomonas as a massive generalist. You can find it thriving in acidic soils, clinging to plant roots, and causing infections inside animals.
But the critical scientific question here is why? What makes a microbe a rugged generalist? If we look at the genomic architecture, generalists thrive in highly structured, aggressively fluctuating environments like topsoils. To survive in soil, a microbe has to endure a drought one week, a torrential flood the next, shifting pH levels and chemical warfare from surrounding fungi. Because of this constant environmental whiplash, generalists tend to have physically much larger genomes.
Right, they literally have to carry a heavier backpack of tools to survive.
Exactly. That larger genome encodes a vast array of utility functions. We are talking about diverse oxygen tolerance mechanisms, the ability to rapidly switch metabolic pathways to scavenge different nutrients, and crucially, an arsenal of intrinsic antimicrobial resistance genes. They evolved these heavy-duty tools to survive diverse existential threats in the wild.
And when we trace that back to human impact, it leads to a genuinely chilling realization. Many of our most notorious hospital-acquired human pathogens score incredibly high as environmental generalists.
Yes. Bacteria like Salmonella, Klebsiella, and various Burkholderia species are highly adaptable generalists. This completely reshapes the public health perspective. It shows that these pathogens aren't just perfectly evolved, dedicated human hunters. They're built to survive in harsh environmental reservoirs like agricultural runoff or stagnant water for extended periods. Their sheer rugged adaptability is precisely what allows them to subsequently colonize a human host, evade our immune system, and resist our antibiotics so effectively.
Let's do our final mini recap here. We saw that microbial diversity peaks at the equator, just like animals and plants. We learned that microbes operate either as adaptable generalists, the backpackers, or highly tuned specialists, the homebodies. So a question for you. Does knowing that the bacteria most likely to make you sick are highly adaptable environmental backpackers change how you view pathogens? They aren't specifically targeting you, you're just another habitat they can conquer.
It's a sobering thought.
It really is.
Yeah.
And it leads us directly into our final segment, the blind spots of science. Despite meticulously processing 2.4 million samples, the most profound thing MicrobeAtlas did was expose the terrifying scale of what we still do not know.
The overarching statistics are humbling. Of all the OTUs, the hundreds of thousands of distinct microbial groups identified in the MicrobeAtlas, a staggering 91% are completely unnamed and functionally uncharacterized.
91%. That means over 9 out of 10 microbes we mapped have no cultured representatives in a lab, no fully sequenced genomes, and no known biological function. They're just invisible entities wearing a 16S ID badge that we can ping on a map, but we are completely blind to what they actually do for the planet.
And that immense lack of knowledge is heavily driven by deeply entrenched sampling biases in the scientific community. When you look at the MicrobeAtlas globally, massive disparities emerge. Over 50% of all the samples in the entire database come from animals. And when we say animals, we aren't talking about a diverse menagerie. We overwhelmingly mean humans and lab mice.
It's a reflection of where the money flows. We care about human disease, so clinical research funding heavily prioritizes sequencing human guts and the mice we use to study human guts.
It is entirely understandable from a funding perspective, but from a planetary perspective, it's incredibly skewed. Contrast that 50% animal figure with the oceans. The oceans cover more than 70% of our planet's surface and serve as the foundational engine of the global climate and carbon cycle. Yet marine environments account for less than 8% of the sequence samples in the database.
The bias isn't just environmental, though. It is starkly geographical.
Very much so. There is a massive, disproportionate oversampling of the northern mid-latitudes, specifically Europe and North America. Meanwhile, incredibly diverse and ecologically critical ecosystems like the global savannas, massive tracts of the Amazon, and vast stretches of the global south are severely undersampled. Now, to be fair, the MicrobeAtlas vastly improves on this compared to legacy projects. It covers about 61% of global latitude-longitude winds compared to the old Earth microbiome project's 15%, but the dark spots on the map are still vast.
We are essentially trying to understand the intricate dynamics of the global economy by exclusively looking at the municipal budgets of a few cities in North America. We are missing the macro picture. But within these biases, there was one specific finding in the paper that really caught my attention because it challenges a core, multi-billion-dollar pillar of medical science. We need to talk about the lab mice.
This is a finding with massive implications for the biomedical community. When the researchers ran clustering algorithms on the metadata grouping samples by how similar their entire microbial communities were, they found that the gut microbiomes of lab mice form a highly distinct, fiercely isolated cluster on the map.
Meaning they don't look like wild animals, and more importantly, they don't look like us.
Exactly. This cluster is heavily separated even by specific lab facilities and standardized chow diets, but most critically, it differs markedly from the baseline human gut communities. Lab mice live in highly sanitized, strictly controlled, artificially sterile environments, and they are extensively inbred.
If I'm understanding the implication here, we rely on these mouse models to test oral medications, study autoimmune diseases, and research the gut-brain axis. If their microbiomes are forming this isolated artificial echo chamber that is fundamentally less diverse and structurally different from a human gut, how can we trust the data? A drug might be metabolized completely differently by a human's microbial community than by lab mouse's.
That is exactly the critical debate this data fuels. It calls into question the translational reliability of mouse models for any disease pathology heavily mediated by the microbiome. If the biological baseline of your test subject is fundamentally alien to the organism you are trying to cure, translating those results from a sterile lab into a messy human reality becomes incredibly fraught with error. It's a profound realization derived just by stepping back and looking at the unified data.
So if we pull all of this together, what does this ultimately mean for us? We've covered incredible ground today. The MicrobeAtlas isn't just a static database. It's an entirely new, high-resolution lens for viewing the biological mechanics of Earth. It took fractured, noisy, siloed data and wove it into a connected planetary web. For the first time, we can trace a single, highly adaptable bacterial strain from a fertilized farm in Europe through agricultural runoff all the way to a river ecosystem in South America. We can literally see the shape and movement of invisible life.
It is a monumental achievement in bioinformatics and data harmonization. The framework they've built here will be foundational for decades of future ecological and medical research.
It absolutely will. But I want to leave you, our listener, with a final, somewhat provocative thought to mull over. We've talked about how this atlas maps the microscopic layer of the Earth. But right now, we also have fleets of Earth observation satellites orbiting above us, constantly monitoring macro-level data. They're tracking shifting global cloud dynamics, the precise height of forest canopies, and real-time ocean surface temperatures.
Imagine a future, perhaps only a few years from now, where we seamlessly cross-link this invisible genetic microbialist data with that macro satellite data. If we know exactly how these rugged microbial generalists and specialized homebodies react to subtle climate fluctuations, could we eventually forecast massive ecological events from space? Could we predict an agricultural ecosystem collapse before a single plant visibly withers, just by monitoring the microscopic shifts in the soil? Or even more pressing, could we forecast the emergence and spread of the next pandemic-level pathogen simply by watching the planetary conditions align for it? The macro and the micro, completely and inextricably linked.
Thank you so much for joining us on this deep dive into the MicrobeAtlas. The next time you walk through a local park, swim in a lake, or even just look at a globe, I hope you take a second to consider the billions of invisible backpackers and homebodies thriving right there beneath your feet, quietly running the world. Keep looking closer at the invisible systems around you. Catch you next time.